Okay, so here we are. It's another edition of Micro Seminar. Today we're going to be hearing from Karrie Weber, who's at the University of Nebraska, Lincoln. She's going to be talking about viruses in unusual circumstances, which will be really, really killer. Karrie did her undergrad work at Texas A&M, and then she went to Alabama to do her graduate work with Eric Roden. And then I met Karrie at Berkeley, where she was doing a postdoc with John Coates. And Karrie was a wonderful mentor, so it's a thrill to have her on Micro Seminar and hear about the new work that she's doing now. So Karrie, I'll let you take it away.
Well, thank you, Cameron. And I'd like to thank both Jen and you for putting this seminar series together. This has been a great idea, and I've learned a lot over the summer and have enjoyed it, and I'm glad to be a part of it. So today I'm kind of taking in presenting something I haven't really presented a whole lot before with respect to viruses in subsurface environments. So I'm just going to switch over here. So what I'm going to be talking about today is some of the work that we have been conducting in my laboratory looking at viruses in environmental systems. And so in the background here you're just seeing an epifluorescent micrograph of viruses, and to start off the talk. And this work, of course, couldn't have been conducted without a variety of individuals in my laboratory. And that is Don Pan, a graduate student, as well as Zhang Huan Tan. They've conducted most of the research that I'm going to be talking about today. So, and I can, of course, have to thank collaborators in which some of the research I'm going to be presenting, and that is Daniel Snow and Yusong Lee here at the University of Nebraska at Lincoln. And of course, some of the work I'm going to be presenting was conducted in collaboration with Ken Williams at Lawrence Berkeley National Laboratory. And we have received some funding from the U.S. Department of Energy, UNL Research Council. Don Pan has been supported by NSF IGERT for this research. And we have piggybacked off another USGS project to obtain some subsurface samples, which I'm going to talk about in a little bit more detail. So much of the work that I've conducted has been looking at microbial life in the subsurface, and I've been particularly interested in those organisms that mediate nitrogen, iron, uranium, biogeochemical cycling. And as many of you know, that microorganisms that are present in subsurface environments are rather abundant, especially when we take into context all of microbial life on Earth. And even in that continental subsurface environment, up to 1/3 of all microbial life can be present there. So I was quite curious in thinking about biogeochemical cycling of how do viruses influence these processes that I'm interested in understanding. So of course began questions and thinking about, well, viruses are so abundant. You know, they represent 10 to the 5th to 10 to the 7th VLPs per mil in these subsurface environments. So they are abundant. And when we start taking into consideration all the viruses on Earth, they're very abundant. And in many environments, viruses will outnumber the microbial hosts and those cells. And they can cause disease, influence the microbial ecology of a system, as well as influence biogeochemistry. And there's been, yes?
Can I just interrupt you? I'm sorry. Can you hide that little thing down on your screen that's showing the Google Hangout so we can see your beautiful slides more clearly? Perfect. Thank you.
Oh, thank you. Sorry about that.
Yeah, sorry to interrupt.
No, that's okay. So there's been much less research that has been conducted influencing and trying to understand the influence of viruses on biogeochemistry. What we do know are those studies that have been conducted in marine environments and in freshwater pelagic systems. And so from there, we began to think about a subsurface system and how viruses might play a role and what they would do and how they would live, or not live necessarily, as viruses don't live, but rather what role they would play. How would they influence those cells that are living in these environments? And one of the complicating factors of thinking about viruses in these subsurface environments is the fact that we have this mineral matrix that's present. So after all, it's dirt, right? It's all these solid surfaces in which can absorb viruses. So these absorption processes are going to be very significant as they're going to influence infectivity. But in these pore spaces where microbial life is going to exist, and or maybe on the outside of those biofilms, which may cover a mineral matrix, you're going to have the potential for viral infection to occur. And various metagenomic studies that have been conducted in the subsurface have indicated that there is the presence of viral genes within these metagenomes, which do suggest that there is a significant influence of viruses in these subsurface environments. And so if we can take into consideration the different life cycles of a virus, or how a virus might exist, it might be from the context of looking at a lytic cycle, where the virus might infect, replicate its DNA, begin to produce viral progeny, and then, of course, lyse the cell. And that, of course, is going to alter biogeochemical cycling. The alternative can be where a virus might infect a cell and enter a lysogenic cycle, where the viral DNA becomes part of the host chromosome, can replicate with the host, or potentially with some environmental stressor that does occur in these environmental systems, lyse the cell and as it enters the lytic cycle. One infection that I do want to mention, because I do think that based on some of our research, I'm going to talk a little bit about this today, is that some of these chronic infections may potentially be significant in some of these subsurface environments, and that very little is known about chronic infection in these types of environments. So what role do they play? So I've kind of highlighted a little bit, but what we do know based on the studies that have been conducted is that viruses do play a significant role in horizontal gene transfer and can contribute to evolution. And there have been some studies that do indicate that metabolic genes can be transferred, as well as new metabolisms or the production of even toxins can be conferred through viral infection. And of course, this does then contribute to microbial mortality. So it results in cell death, and it can control microbial community structure as a result, as populations can be lysed and allow for a new population to take over as the predominant organism in the system, or organisms for that matter. And finally, the biogeochemistry. If we start to think about, after all, viruses are lysing a cell, so they have the potential to begin to release dissolved organic carbon as well as particulate organic carbon in cell debris. So there is this potential of freeing up available carbon, as well as nutrients such as nitrogen and phosphorus, and maybe in some environments, metals that might be needed for cells to take advantage of in that environmental system. But we really don't know what the significance of this process is overall. So we simply began asking that question. Well, what role do viruses play? And of course, are they even significant in these environmental systems? And we're interested in my laboratory on metal mobility. So we, of course, always are thinking about how this might play a role in metals. But the first story I'm going to tell you about is just simple. And one is our first objective was simply to demonstrate the production of viruses in subsurface systems. Is it significant at all? So that is the first question that we wanted to ask. And then finally, we were interested in just that virus as a potential surface for metal absorption. And so, I'll tell you just a little bit about that today in the context of some biogeochemical cycling. So first, I'm going to talk about work that has been conducted by a graduate student in my laboratory, Don Pan, in looking at the production of viruses in subsurface sediments. And we focused on a site within the High Plains Aquifer here in the state of Nebraska. So this was a site that has been researched by other investigators, my colleague Dan Snow at the University of Nebraska at Lincoln, to study nitrate and uranium. And we're continuing to study nitrate and uranium in this aquifer as well. So this is a shallow alluvial aquifer. The water table at the time of the samples were collected were at 20 feet. And we do have kind of a clay aquitard that does exist at about 60 feet. So it's a rather shallow system, easy to collect samples with the geoprobe, which you can see here in the background. And based on prior research that had been conducted at this site tends to be very it tends to be a reduced environment with a reduction potential below zero. So we wanted to preserve those anoxic conditions in this system. So immediately upon core collection, the samples were preserved under an anoxic headspace with argon. So we just filled some tubes and sealed them up with argon and placed the cores inside these tubes so we could take them back to the laboratory to process in an anaerobic glove bag. This aquifer, given that it is a shallow aquifer here in the state of Nebraska, we do have a lot of agricultural production, and thus we do have significant and extensive nitrate contamination throughout the state. This site is not immune. There is a significant amount of nitrate in the system above the maximum contamination limit. So we have about 30 ppm in this system, which here you can see the amount. And specifically, we were interested from the context of studying viruses in the system. When we wanted to set out to look at viral production, we were first interested in simply demonstrating that we could produce viruses. So we wanted a simple system in sediments. I don't know if there is such a thing, a simple system within sediments, but when we take into consideration that if we can add different products and reactants that are going to remain soluble and be converted to gas, that at least simplifies the system. So thus, this is why we chose to work with a nitrate reducing system. We selected the region to study, and we did do most probable number enumeration analyses throughout the 40-foot section of the core that we collected. And we specifically decided to focus on this lower region here from 58 to 64 feet, simply because this is where the most abundant nitrate-reducing microbial community was present. And just to point out that likely that one of the reasons this tends to be a little bit more abundant is simply because we're going from sand gravel down to clay in this system. So this is abundant likely because this is where you have sand and gravel meeting this clay interface. So there's a ample opportunity for the exchange of dissolved organic carbon as well as nitrate electron acceptor in this simple system, or this aquifer here. So we have about 10 to the 10 cells per gram of sediment. So this seemed like a great area to target to begin to look for virus production in this system. So we selected this depth as well as another depth to create these large sediment slurries that we needed in order to collect the amount of sample for processing. We needed a large volume, so we ended up combining two of these sections in order to generate this sediment slurry. And we simply created an enrichment, and we envisioned simply using a nitrate-reducing enrichment culture system. So we added electron donor. We selected acetate simply because it is a non-fermentable substrate. We used 13 seed-labeled acetate so that we could quantify how much carbon dioxide was produced from the as a result of the mineralization of acetate in this system. And then we selected nitrate as the electron acceptor for respiration. And given that we had an abundant nitrate-reducing community, we expected to have nitrate reduction occurring in this system, and we did. And within this sediment slurries, as we would expect, we see that nitrate is reduced coupled to carbon mineralization. So in this upper graph here with nitrate on the primary y-axis, nitrite is located on the secondary y-axis. Nitrate is denoted here in solid squares. Nitrite is denoted by the open symbols. And nitrate and acetate, you're going to see this treatment. Here is the circles. Nitrate only is the squares. So what you see is as nitrate is reduced, nitrite does accumulate, and this is concurrent with the consumption or oxidation of acetate, as we see the production of CO2 in this study. We do see the transient accumulation of nitrite as a result of this experiment, and for about 24 hours, we don't have a significant, there's some nitrites that reduce, but not a lot, and that we can see in carbon mineralization, so there is some lag that does occur between the two of these before nitrite is reduced again, and this is through denitrification, as we didn't measure ammonium production in this study. And we did see that we do see an increase in dinitrogen concentrations in this study here. So from this, we do know we had a nitrate reducing microbial community that was indeed induced. And this is what we would expect based on all previous studies in subsurface environments. We started to look at the microbial community using Illumina sequencing. So we have about 50,000 to 100,000 reads that make up each of these community memberships for these different time points. And from the initiation of the experiment, we do see a transition from domination by the beta proteobacteria to the gamma proteobacteria tending to take over or dominate this nitrate reducing enrichment system. Now, of course, we don't really expect that. Well, we don't really know, and I haven't described yet, the role that viruses are playing. But when we started to look at those viruses in this system, we started to see the accumulation of viruses. Now, what I'm going to describe as a virus, for those out there that may not necessarily be familiar with the technique, is simply that we are using, apologize for this, we are using viruses that are smaller than 200 nanometers. So this is any particle, rather, that is smaller than 200 nanometers in size, is DNase resistant, and can be stained with a nucleic acid. We specifically chose cyber gold, or cyber green in this case, to use as our nucleic acid stain. Cells are anything that is above 200 nanometers in size, and we did stain all cells with acridine orange for enumeration in this case. So what we end up seeing overall is a significant increase in this virus to cell ratio over the course of the study, and this is concurrent after we see the reduction of 10 millimolar nitrate in the study, and we have a significant increase. 10 to the third is our ratio that is a result in this study. So we know that viruses are being produced. So we were successful in achieving our objective of producing viruses. But after all, what does this mean? I mean, we would expect that if you're inducing nitrate-reducing bacteria in this system, that viruses would be produced. Well, we've demonstrated that, but we were particularly interested in how that might actually be correlated to carbon production in this system. So when we begin to look at that change in virus abundant versus the amount of acetate consumed or an amount of CO2 produced, we do see this positive correlation. More interestingly, when we start to look at this change in cell abundance versus the amount of acetate consumed and CO2 produced, we don't necessarily see a significant correlation with the number of cells. And at first that did surprise us, because we would expect that cells are the organisms that are responsible for nitrate reduction in this case. We would expect that the cells should be actively metabolizing. But what we end up seeing is that we don't have that statistically significant correlation. But if we think about viruses are being produced, what is the effect of the virus in this case? It is to lyse the cells. So perhaps what this data is actually telling us is that the cells are being lysed as a result of viral infection in this study. So viruses lyse the cells. We're not actually seeing an increase in cell abundance because of lysis. But viruses do remain in the system. We don't know what their decay rate is over the course of this study, but we do know that there is viruses that are being produced. Of course, they're not necessarily going to be subject to the same decay that a cell would, especially undergoing some predation pressure that the cells are. So this was interesting because what this allows us to begin to think about is perhaps maybe using some virus to cell ratios might be an indicator of metabolic activity in these environmental systems. And of course, we have to think about, well, if viruses are correlated to carbon consumption, we would expect that there might be a change in microbial community structure, which we know there was a change in microbial community structure. So we were curious as far as, do we see a correlation in this change in community structure as a result of this, the change in viral abundance. So if we look at the change in relative abundance of the gamma proteobacteria and the beta proteobacteria versus the viral abundance, what we end up seeing is that we have a negative correlation with the beta proteobacteria, which suggests that they may be potentially lysed by the viruses, or we have an increase and we have a positive correlation with the gamma proteobacteria, which may indicate that they're not necessarily being lysed by the viruses produced in this study that we're measuring. So this is pretty interesting. However, correlation itself in this case may not necessarily say that yes, viruses are infecting and influencing these organisms, suggesting it is a possible outcome. As the enrichment itself could potentially have an effect on what is happening in the system. But what was most interesting, and I guess I overlooked and forgot to state here, when we actually looked at the community membership within the gamma proteobacteria. The gamma proteobacteria is almost virtually all Pseudomonas species. And we were interested, too, if we could actually isolate one of these dominant organisms from this environmental system. And from the most probable number enumeration series, we did indeed isolate a Pseudomonas species, all the 10, from these sediments. And actually, we have several different isolates, and all of them ended up being a Pseudomonas species that we have obtained from the enrichments and isolations that we have achieved. And we were quite curious if whether or not, one, we could infect it, and we were unsuccessful trying to reinfect the Pseudomonas isolates with the pore waters that we had obtained from the experiment. So what Don ended up trying was simply doing a test of the pure culture itself for the ability, or to test whether or not it actually did harbor a lysogen inside. And what often is done here is using mitomycin C, which is a DNA cross-linker, which will, of course, induce DNA damage and thus the SOS response, which should induce the lysogenic phage to enter the lytic cycle. So we should see the production of bacteriophage as a result of the treatment with mitomycin C. And indeed, that's what we saw. However, after looking at viral particles via transmission electron microscopy, what we end up seeing is a single-stranded DNA phage that is present, or, well, I say single-stranded DNA, but a filamentous phage, and most often they are single-stranded DNA phage. And single-stranded DNA phage aren't known to cause a lysogenic infection. So we had to go back to the drawing board. So perhaps this data necessarily doesn't indicate that this was a lysogenic bacteriophage, but rather it's just indicative of cell lysis. So an additional experiment was conducted in which chloroform was added in the place of mitomycin C and compared with the results from mitomycin C. And indeed, we actually did have the same amount of lysis, which did suggest that it's actually cell lysis that's occurring that releases the phage from the host in this case. So what that tells us is that perhaps this isn't necessarily a lysogenic infection, but rather a chronic infection. So, you know, we don't know necessarily if there is some advantage of that chronic infection does confer to this Pseudomonas species in this environment, and of course does begin to pose an interesting question, especially towards microbial community ecology. And what's interesting is if we actually go back to this aquifer system and we look at the distribution of organisms throughout this aquifer and up in this sand and gravel portion, we find that 98% of the microbial community is actually Pseudomonas species. So perhaps our enrichments that we conducted in this study aren't too far away from the real story, at least in the sand and gravel portion. So there's a little bit more, quite a bit more that we would have to do to be able to understand exactly what's going on and what might actually promote Pseudomonas to become the dominant organism here. But one thing that I think we need to start thinking a little bit more about is how viruses might play the role in the outcome of this microbial community structure as a result of these studies. So this study, what we demonstrated was that biostimulation does result in the production of viruses, and that we can observe a correlation between carbon mineralization and virus production in these environmental systems. And one thing that this study opened our eyes to was also that we need to think more carefully too about these single-stranded DNA viruses, as well as potentially RNA viruses, when utilizing methods to begin to look at the viral community. And I will admit that we did try some techniques to begin to look at the viral community. Unfortunately, all of our purification techniques, as well as our molecular techniques, were geared towards double-stranded DNA. So we ended up overlooking what would have been based on the enrichment study, a single-stranded DNA virus. So I think these organisms that might be infected with these single-stranded DNA viruses could potentially be important in some environments, and so that's just something to carefully think about. Well, so from the results of this study, we demonstrated that in a bottle we could produce viruses. So we were curious whether or not we can actually translate our laboratory research to the field. So in collaboration with Ken Williams at Lawrence Berkeley's National Laboratory, and utilizing the resources and the site available in Rifle, Colorado, we piggybacked on another experiment that was going on out there, and this was an in situ stimulation experiment. This is a little bit different than prior experiments that had been conducted out at Rifle. Mike Wilkins discussed some of the acetate amendments into the Rifle field site earlier this summer, which are important, and I'll describe that here in a second. But just to provide you, everyone, with an overview of the Rifle field site located in Colorado, is that it was once sitting below a uranium mill. So a lot of ores were processed out here and thus led to contamination of uranium as well as other heavy metals as well at this site. So it has since been remediated. The topsoil was removed, replanted, but yet within the groundwater there does remain some of the heavy metals that were present as a result. But regardless, it sits right next to the Colorado River, and so it's on this floodplain. And it does represent an alluvial aquifer. And one of the advantages of the prior research that has been conducted out there, and even the experiments which Mike Wilkins described earlier this summer, where these groundwater systems and these subsurface systems were amended with acetate, created this reduced zone. And so it's naturally reduced out there, and there are a lot of hot spots. And in many subsurface environments, especially in these aquifers where you have hot spots, and these may be where clay lenses appear and high organic carbon might become trapped in these environments, and you have hot spots. And it's hard to detect some of these natural hot spots, especially if you're trying to do an experiment to find out what is the response of inputs into these naturally reduced zones. So this experiment was essentially designed to take advantage of these prior experiments that had reduced. So we can call this a simulated naturally reduced zone. And so this is an image of the well field, and so you can get a better idea of what it appeared. These wells denoted here by these larger circles would have been injection wells. Within the well field, groundwater flow would be in this direction denoted here. And acetate would have been injected in these solid wells 2 years prior to the beginning of this experiment. And for this experiment, what was done was the addition of oxygenated groundwater. So oxygenated groundwater was injected. So there was water removed from another location that would have been upstream of the initial experiment, and aerated and then re-injected back into the subsurface. So this would be the oxygenated groundwater that was injected. So now to orient you with the sampling locations, CU upstream, this is going to be the upstream which wouldn't have received any amendment. CD11 was wouldn't have received an amendment from the oxygenated groundwater and would have had a past, would have been affected from past bicarbonate injections, but in a sense, in this time, it would have served kind of as another control in a downstream location. And so CD18, CD01, and CD04, these would be the downstream wells in which we'll see some data presented from here. So Don Pan conducted this research, and again in collaboration with Ken Williams and a technician, Mark Robbins, out at the Rifle Field site. So one thing I do want to point out, we're going to slightly change the definition of what we're calling a virus in this case. And that's simply because, based on some of the research that Kelly Wrighton had conducted out in Jill Banfield's lab, is that they identified some bacteria that were smaller than 200 nanometers. And so in this study, we wanted to make sure that what we were enumerating as a virus was definitely called indefinitely a virus. So we simply chose the cutoff of 100 nanometers in this case as a virus. Now, we have done enumeration studies to look at the different fractions between 200 and 100 nanometers, and they're not statistically different than each other, but there is that 200, less than 200 fraction tends to be a little bit higher than that less than 100 fraction. So we want to, in this case, we want to be certain that we are calling these viruses. So we're just going to simply use this cutoff here as 100 nanometers. And the same technique applies where a virus is a DNase-resistant particle that will stain with a nucleic acid stain in this study. So now there's a lot of data that I'm going to be presenting here, so bear with me. But it all comes back down to this reduced to this reduced zone. All the wells here that would be in response and downstream of the oxygen injection are denoted in this column right here. The background wells, this would be the upstream wells, the upstream well, CuO1, is denoted here, and the downstream well that wouldn't necessarily have received the input of oxygenated groundwater is CD11. So one thing that you'll note, whether we look at the simulated naturally reduced zones or the background wells, is that we do see a decrease in the oxidation and reduction potential, which is counterintuitive. We would expect that with the input of oxygenated groundwater, we should see an increase in that oxidation reduction potential rather than a decrease in the ORP in this case. But we even see the same decrease in the oxidation and reduction potential in the background wells. So something else is going on here that might be separate from the injection of oxygenated groundwater in this case. But one thing is also clear is that even with the injection of oxygenated groundwater, we don't see significant increases in dissolved oxygen concentrations, which suggests that it's being immediately scavenged as soon as it is put into the system. So this, again, it's curious that we see this significant decrease in this oxidation reduction potential. Again, as I mentioned, we do see it here with the upstream location as well. So perhaps this may suggest that there are some inputs of dissolved organic carbon that are coming in naturally into the system, which might help drive down that ORP as a result. Another explanation may be that inputs of oxygen can also help catalyze further degradation, decomposition of organic matter, and also freeing up some dissolved organic carbon. And we're going to come back to that shortly. But one thing that we do observe as we see this decrease in this oxidation and reduction potential, we also see this increase in virus abundance. And that seems to be consistent regardless of which well we're looking at, whether it's this wells that would be impacted by the oxygen injection or even the background wells, where in theory should be unimpacted by the oxygen injection. So we are indeed seeing an influence here of these microbial metabolic activity, and perhaps that's indicative of this decrease in ORPs, which we would expect as these organisms continue to respire, and as a result their metabolic activity is increasing, as thus then we're producing viruses in response. So what we're seeing is consistent with what we saw in the bottle experiment that I had previously described, is that we are indeed seeing this production of viruses in response to some perturbation or microbial activity in this system. But similarly, we don't see a change in total cell abundance. So here, the graphs here are oriented slightly differently because the upstream well is in this row, and the CD11, which is the downstream, another control well located here, and the simulated naturally reduced zones are denoted here. And we do see some slight increases in cell abundance in some cases within the simulated naturally reduced zones. However, we don't really see those significant increases within the upstream locations, whereas we do see these fluctuations again with the virus to cell ratio. So further support, which does suggest that perhaps maybe we can consider using virus to cell ratio as an indicator of metabolic activity, especially when cell abundance may not be changing in these systems. But it also makes us think that perhaps when we're thinking about the organisms that might be driving some of these biogeochemical cycles, that we're missing many of them because they could be lysing in response to viral infection. So one of the things is we started looking a little bit closer at our data and some of the challenges that we face working in this subsurface environment and relying on groundwater sampling in order to collect samples to do virus enumeration. What we end up seeing is after 25 days, we have had a significant decrease in ferrous iron concentrations, and we would expect that based on iron oxidation. So abiotic as well as microbial processes that would facilitate iron oxidation and thus iron oxide precipitation. And if Ken Williams placed a camera down the well, and what you're looking at is well of CD01 before it was pumped at the beginning of the experiment. And you can see that it's gray, there's really not much color to it, but by the time the experiment ended, iron oxide flock ends up coating the interior of the well, which demonstrates that there was a significant amount of iron oxidation in response to the injection of oxygen into this subsurface system. So this could have an effect in pulling VLPs out of the solution. So this does complicate things, especially when we're starting to think about how we are interpreting some of our data in this experiment. So when we started thinking about how are we going to look at some of the organic carbon data, because we're a little bit curious, especially since we saw that decrease in the oxidation reduction potential, we're going to focus and look at the early portion of this experiment where we didn't have, well, we know that there's iron oxidation occurring. So some viruses could have been pulled out of solution at this case, but we didn't have this significant precipitation. And we have collected other samples, which we've concentrated via tangential flow, in which after about a month, we began to see clogging of the tangential flow filter as a result of iron oxide flock, and had to start dealing with strategies to clean up the filter itself. So that's one more kind of qualitative description, and we know a little bit more about the precipitation of the iron oxides in this study. So when we start looking a little bit closer at the amount of dissolved organic carbon in the system and focusing on these first 25 days of this experiment, we do see these fluctuations in dissolved organic carbon, much less within the upstream location of this well. But we do see this increase in this virus-to-cell ratio. So perhaps that virus-to-cell ratio is reflecting some of that microbial metabolic activity that is ongoing as a result. And in response to the wells that had received the prior acetate amendment, now the oxygen injection into the system, there is significantly more dissolved organic carbon in this system. Now, this in the release of this dissolved organic carbon in the system could be a result of cell lysis, but it could also be the fact that if we're adding oxygenated groundwater into this previously reduced system that was high in organic matter, that we may simply be increasing decomposition of that organic carbon in the system and increasing some of the bioavailable dissolved organic carbon. And so that's yet another study that will have to be done in order to have a better idea about what is actually happening in this system exactly. But if we were to look a little bit closer at this, I think that the take-home is that stimulation of a microbial community will, in situ, also will result in the production of viruses. So viruses are being produced in these subsurface systems. So it's what we saw in our enrichment experiments. We're seeing some very similar patterns. Now, granted, we haven't replicated everything yet from that initial enrichment in the subsurface, but we are seeing some parallels, which we have observed in the bottle experiments. So this is carrying into the natural environment. So it's something that we do need to take into consideration as we begin to consider what are the impacts of viruses on biogeochemical cycling. Now, I don't have too much more time to discuss the work that we've been doing looking at the absorption of heavy metals and radionuclides to viruses, but I want to highlight some of the research that would have implications towards starting to consider viral infection in these subsurface environments, because there is a parallel here. So this work was conducted by a master's student in my laboratory, Zheng Tan, who just graduated this past May. So the objectives of this experiment were simply to take a model bacteriophage, add it into an aqueous matrix to begin to measure absorption of a metal to the surface of this bacteriophage. So a series of experiments were conducted by Zhang, which had demonstrated that indeed viral surfaces are going to absorb the heavy metal. Now, what was most interesting is that when we started to look at the infectivity of the adsorbed zinc to the surface of the virus, we noted that there was actually an increase in infectivity in those viruses that had zinc adsorbed to the surface. So what you're looking at here is the number of plaque-forming units. So this would be the number of infectious units or infectious bacteriophage that were added into the system. So if we keep and remove the zinc from the viruses, and so that we have no zinc present, we'll use this as a control. Additional controls included the addition of 150 micromolar zinc, as well as EDTA, to serve as a chelator to keep the zinc from binding to the surface of the bacteriophage, as well as the addition of salt. And indeed, what we see when we have zinc alone is we see more plaque-forming units that are formed as a result. And this is consistent even after 20 hours of the incubation. So we do know that there is some increase of infectivity. So what is actually happening here may simply be twofold. One could simply be that the zinc on the surface is help preventing decay, or perhaps the zinc on the surface is assisting in promoting absorption of that phage to the host cell, which is increasing infection. But nonetheless, what it indicates is that that aqueous chemical matrix that's going to surround a virus can potentially have an impact on infection. So taking viruses into account in predation models are going to have to also include what is the reactivity of these viruses once they have been influenced or metals are absorbed to the surface as well. So in conclusion, we've have enjoyed starting to begin to ask some of these questions, and it's been a lot of fun to start thinking about some of these systems. To think about how carbon biogeochemistry might be linked to viruses in these subsurface environments, and what our results have indicated is that this virus to cell ratio can reflect there's host metabolic activity within these subsurface environment, where if we were to measure just cells alone, we might miss out on some of this activity as a result of just cell lysis. And of course, in this aqueous matrix, in this milieu, is that viruses can absorb heavy metals, and we also have to be cognizant of the fact that mineral precipitation can also pull viruses out of solution. And also that whether it is absorption can influence viral infection of these hosts in the subsurface environment. And what I didn't have noted on here, but also the inverse of thinking about mineral precipitation also would have a negative effect, right? If the virus itself is coated in an iron oxide, potentially that's going to have implications as well. So, there's a lot more that we have to do to conclude, and we're going to be continuing to study viruses in the subsurface environment and how it relates to biogeochemical cycling. So with that, I'd be happy to take any questions, but before I move to the question and answer session, I just want to point out that we're looking for interested postdocs and graduate students to look at other projects going on in our laboratory, considering the dissolution of carbonate minerals and methanogenesis, as well as nitrate-mediated metal radionuclide oxidative dissolution. And thank you.
Thanks, Karrie. That is a really cool talk. There's a lot going on down there. I think that you got some. There was definitely some attention on the Twitterverse here in terms of live blog or live tweeting that talk, which is always a good test of interest. There's a question here from Maya Breitbart. Maya, forgive me if I'm mispronouncing your name. I've actually never heard it. I've only seen it in text. But her question is back to those original filamentous phage that you were talking about. Are they large enough or bright enough to see with cyber?
So we have been able to actually see them with cyber green. So when Dawn had conducted some of those initial studies to look at the mitomycin C studies, where we're looking to determine originally if it was lysogenic or not, that was all done with cyber green. So we can actually see them with cyber green. But when we've done DNA extractions, DNA that has been extracted has been challenging to see with cyber green. So we've had to use cyber gold with the DNA extracts.
Okay. And another question from Maya. Any idea where, why, or how the phage bind metals? And do you think it's specific or non-specific? And then she also comments, "I'm so excited."
So well, that's a great question, and you know, we started to look at some of the reactive proteins that could be on the surface. You know, and starting to think about some of the parallels. There's a lot of research that's been done on bacterial surfaces, serving for just as absorptive sites. So why not viruses? Why not viruses? And a recent research has been published too about viruses serving as nucleation sites for mineral formation. So, you know, there's no reason that viruses can't be playing a role in mineral precipitation. You know, and they're not represented in the fossil record or haven't been studied in the fossil record either, right? So, but they should be there. So there's a lot of possibilities that are out there, and that's going to depend upon the surface of the virus itself and what reactive sites. Do we know where the zinc is binding? Not exactly. You know, and we simply started to use zinc because it's an easy metal to work with. But you know, now we have to start testing other bacteriophage and other viruses.
Do you think that you know a lot about metals and metal crystalline formation. Do you think that there's going to be certain metals that would be more conducive to crystallizing or nucleating on viruses than others? And what might be those kinds of properties?
Well, you know, it's going to depend upon the reactive sites that are going to be present on the surface. And of course, the coordination, right? So zinc might be rather small. So iron could absorb. And there are a few studies that are out there that have shown iron oxidation and iron oxides that can actually precipitate and coat a cell. A recent study demonstrated with some phage where you could have calcium carbonate precipitate on the surface of viruses as well. So it is going to depend upon the nature of the metal itself, whether or not it's what type of mineral is it going to form, what nucleation site it could be. So
yeah, that's really exciting. Are there any questions from inside the Hangout? If you guys have one, just unmute your mic and ask away. I have sort of a... Oh, go ahead. Go ahead, Tyler.
Yeah, this is Tyler. This is might be a bit of a softball question for Karrie, but just, it's kind of big picture, but it really, you know, in the last several months, you hear more and more about the viruses and what they're doing in these systems. So it seems most prudent, like given what we know and especially don't know at this point, to like start considering these as a fundamental part of the community, you know, not the microbial part, but and develop, you know, continue to improve the methods to detect them basically in most studies to understand how they're affecting the processes. So I just want to hear like if you agree with that, and also like what maybe are the methods that we could work on right now to get to that stage. So yeah.
So, you know, that's a great question, and you know, I do think that they are fundamentally important. You know, they are being produced, and they are lysing cells. And and when I reflect upon some of the enrichment studies that I've conducted in the past, just to try to identify which microorganisms are playing an important role for a specific metabolism, I'm using the techniques that I used here to look for the production of viruses. And so I have to wonder, who have I missed? What bacteria have I missed as a result of using these enrichment techniques over time? So one thing, maybe we should also think about how do we measure that lysed community. There's a recent study coming out of Matt Sullivan's lab using some viral tagging techniques. But using that viral tagging technique where you have to have a host, you have to be able to culture the host, right? So it still becomes a challenge. So maybe we need to think about how do we follow that change in microbial community structure, or those microorganisms that are actually being lysed as a result of viral infection.
Yeah, and actually, just a quick, it relates to one other thing I was thinking about, and that is, are there typical responses besides essentially exploding, like as far as RNA or protein production that are known or identified, as far as like these cells are being heavily attacked by certain phage?
Well, I think as we begin to learn more, and so the viral metagenomics that is ongoing is going to be helpful to begin to identify some of those characters, right? Because after all, those are where those genes are going to be. And one thing that I haven't mentioned is these chronic infections. What is the role of the chronic infection, and how could it potentially impact the cell? So I was talking about lysed cells, right? But we also have to consider those cells that might be chronically infected with a virus as well.
Cool. Yeah. Thank you.
Great. Any more questions out there? I just have one, unless somebody interrupts me. I have one sort of like also a big picture conceptual question. We've seen some evidence of these metabolic genes being transferred, or at least being present in phage, and we're very concerned with all that nitrogen in your soil down here in the Gulf, because it all winds up down here. And I guess I'm wondering, are there efforts, or is there a dialogue at all about using phage to potentially transfer systems that might do something good, like nitrate reduction to ammonium instead of denitrification or nitrate reduction to nitrite, for sort of bioremediation purposes? I mean, is that something that this is a little off topic, I guess, but your discussion at the beginning of your talk got me going on that. I just
well, yeah. So you're talking about cleanup of quite extensive system, right? I mean, that's a challenge. And even from the context of groundwater quality and drinking water quality. So most of the state of Nebraska, most of it, uses groundwater as drinking water. And so if you recall from that map, we have a bit of an issue, right? So and water managers are facing challenges, but yet it's the alternative is to clean it up for drinking water and for treatment. So there isn't a lot of in situ work that goes on.
Mm-hmm.
And as far as strategies using phage to confer some other type of process, I don't know how much we really know over trying to force and regulate DNRA, which is what you're suggesting. And if we really, as far as reduction, oxidation potential, how low do we have to go in order for DNRA to become the most important process?
Okay.
So there's some fundamental metabolic questions I think we have to know a little bit more about. But potentially, right? Why not?
Cool. Okay, is there anything else out there? Anybody dying to ask? Remember, one of the great features about this is that if you think of something later, you can contact Karrie. She's on Twitter at @RockinMicrobe, which is an epic Twitter handle, and you can always catch bits of this. We'll have it archived on our website if you want to go back and review anything a little bit later. Okay, so thank you, Karrie. I really appreciate it. This was an amazing seminar, and thanks for contributing your knowledge and expertise to our seminar.
Well, glad I could do it.
Okay.
Thank you guys again.
Thanks, Karrie.
Right.